This morning, we're going to finish up this chapter looking at the last of the five special senses, and that would be a vision. Of course, that takes place in your eye. So we're going to learn all about the eye. Um, and when we do, there are some accessory structures before we get to the eye itself that I want to go over. You can see them here. And so if you'll just stay on this page, we'll move on to the images that show this. First of all, on the eyelids, you probably can figure out why you have eyelids. Can you tell me? Protect your eye, so you're blinking constantly, and that's protecting your eye from impurities that are in the air. There's eyelashes that help with that, but there's also some glands. Do you see this thing called a tarsal gland? Okay, that's within the eyelid, and it produces an oily secretion. Do you see in your nose it says it's a type of sebaceous gland? So it's producing an oily secretion that helps lubricate the front of the eye so it doesn't stick to the eyelid. Okay, so that makes sense. If you've ever had a sty, that's a clogged tarsal gland. You ever had one of those? It's just like a big knot right there, kind of like a whitehead there in your eyelid, but they can be kind of irritating. Just they don't normally get infected, but anyway. Then lining the eyelid, if you look where I'm drawing. All right, this is a mucous membrane called the conjunctiva. It lines the eyelid and it goes up onto part of the surface of the eye. All right, so you see it there, and then you see it all along here. Okay, and because it's a mucous membrane, it produces a mucus type of secretion, which is thick and sticky, so that if anything does get through the area protected by the eyelids, then it can catch it. Exactly, it can catch it. If somebody has conjunctivitis, that's inflammation of that membrane. And so it produces a lot more of the mucus. Like pink eye is an example of conjunctivitis. And the reason it's so pink and red is because there's a lot of blood vessels associated with it. And so those, during any kind of inflammation, remember blood vessels dilate, right? And so you just will see that a lot. Okay, so that's enough on that slide. Right. Some other accessory organs include the lacrimal glands. The word lacrimal refers to tears. A lot of people are under the misconception that your tear glands are right there. That's not your tear glands. Here's your tear glands, or lacrimal glands. They're on the lateral side just above the eye, and they are producing tears that wash over the surface of the eye that then drain into these ducts that you see that lead into the nasal cavity. As far as your test is concerned, I don't care that you learn all of those terms. I just want you to know the lacrimal glands, and I want you to know that they drain into the nasal cavity. Do you remember in A&P1 when we studied the skull? Do you remember on the front of the medial surface of the eye socket, there was a little tiny bone that had a hole at the bottom of it? What was the name of that bone? Lacrimal bone, right in front of the ethmoid within the eye socket. And that's where those ducts are draining through to get into the nasal cavity. Okay? You're constantly producing tears to wash impurities off the surface of the eye. Another reason that you make tears is so that it's always wet on the front of the eye, because we're going to see that the cornea, which is the clear window at the front of the eye, it gets its oxygen directly from the atmosphere and not from the blood. All right, and so in order for oxygen to diffuse across, it's got to be dissolved in water. All right, and so that's another reason it's important that the front of your eyes is always are always moist. You don't notice that you produce tears all the time unless you have that dry eye syndrome where you're not producing them, and then it feels scratchy quite often and real dry and irritated. But normally the tears are just draining into the nasal cavity. If you produce a whole lot of tears, they'll come out onto your face. It's interesting that chemically you got two kinds of tears. You have everyday irritant tears, which is hopefully what you're producing right now. And then you have emotional tears. And chemically, the emotional tears contain some proteins, some albumins, that their levels get elevated when you're under stress. And so this is relieving that. It's removing some of those stress causing chemicals. So that's why sometimes it feels good after a big cry, doesn't it? Whether you're tearing up because of good emotional things. Sometimes we cry because we're happy, but it feels good after you do that. But sometimes you cry when you're really sad. But after a really good cry, you've relieved a little stress because it's removing some of those chemicals from the body. That's really interesting to me. All right, and then another accessory organ of the eye would be the skeletal muscles that control the movement of the eyeball. Humans have 6 of these. You can see their names listed there. The ones that are the rectus muscles are the ones that go straight onto the eye. You've got superior, inferior, medial, and lateral. And so they help the eye with the movement up and down and right and left. But we also have some oblique muscles. This is the superior oblique. You can see it way back in there. But do you see how its little tendon goes across the surface of the eye like that? So when it contracts, it rotates the eye a little bit. So like when you roll your eyes, this muscle helps with that. Some animals don't have that. Like a cow only has the 4 rectus muscles, so he can't roll his eyes to look way over there like that. So he has to turn his head to do that. He just has the rectus muscles. We also have an inferior oblique. You can see it right there. As far as your lecture test is concerned, I don't care that you learn the names of these muscles, okay? But you will have to do that for lab. So I've just got it in your notes here just to give you a heads up in lab when we cover the eye in lab. You just need to know that they're the extrinsic eye muscles. They're skeletal muscles, and they control the movement of the eyeball. We talked about these way back in A&P 1 when we were talking about different muscles that had different sizes of motor units, and these were some that had very small motor units, so you have real precise control over them. And then when we talked about red muscle fibers and white muscle fibers, which ones were the fast twitch and which ones were the slow twitch? Do you remember? The white was the fast twitch, but the downside is they tire very easily. These muscles have a lot of those fibers. So you can move them very quickly, but like when you're reading or when you're looking at the computer screen or through the microscope, you get tired easily, don't you? And it's because you have a lot of fast twitch. All of that, you don't have to know that for this test. You just need to know the extrinsic muscles move the eyeballs. Okay? So I've spent too long on those, I'm sure. But here you can see each one laid out for you. So when you study for your lab, you might want to go back to this online. I know you can't see it with the printed notes so well, but if you look at this online, you can see each of these 6 muscles. All right, now we're going to look at the eye itself. And it's one of my favorite organs. It's real interesting. I want you to notice that there's a lot of structures within the eye. There's also a couple of different kinds of fluid within the eye. One of the types of fluid is real watery. Another one is thicker, more like a loose jelly-like material. And then there's a lot of physical structures, or a couple of physical structures, that light waves have to pass through. They've got to come through the front of the eye. They go through this cavity, and then they go through the lens, and then the light waves go through this cavity, and these cavities are filled with those fluids. Every time light waves hit a different medium that a light wave can pass through, it's going to bend. And the whole purpose of a lot of these structures and fluids associated with the eye is to bend the light. And because when you get to the back of the eye, we're going to name all these parts in a minute, but do you see this little area at the back of the eye, this little indentation right there? Right in the very dead center of that is, if a light wave hits it, you're going to see it in real sharp focus. Okay, so that's your area of sharpest focus. You don't have to even write that down yet. Right. We're going to see at the back of the eye, you've got all the photoreceptors all along here on this area called the retina. So it's all along here. but the ones that are right there are the ones that give you the sharpest focus. And so when you look at something head on, the whole goal is to get those light waves bent so that it all comes together right there at that little point. And that's the goal, is to get it at that point when you're looking straight on at an object. Everything in your peripheral vision is going to be hitting all these other places. You won't see it as sharply focused. Okay, it's pretty interesting. Right here at the back of the eye, this is the optic nerve. If you looked at this picture in your book, it probably gives you an indication. It probably says what kind of section this is, and it may say it's a does it say on that picture, Danielle? A transverse section? You see how it says that underneath the picture? All right, so that means this is not a sagittal section, like up and down. This is like you're looking at it from the top or from the bottom, okay, which is obvious to you on this picture. So it's that type of section. So do you see that the optic nerve projects medially? So it's not at the very, straight back. It angles a little bit. This is the area that's right in the very, just straight back from it. But anyway, there's your optic nerve, and the place where it connects to the eye in the back of the eye, that's called the optic disc. Okay. All right, so as we look at, my marker's not working. Let's see, get it right. Here we go. Okay, so as we look at all these different areas of the eye, we're going to start here. You can see a view of the there's basically 3 layers at the back of the eye. On the left side, there's the outer layer, and then in the middle picture, you can see a real thin, see that real thin little blue line? That's part of the middle layer. And then here you can see the inner layer. And this inner layer is where your photoreceptor cells are found. Okay, so if we name all of these parts, I'll come back. I've got these in the wrong place. I'm sorry. Let me go to the layers, and then we'll come back to those sections. Okay, if we look at the outer layer of the eye, there are 2 structures associated with it. Let me get to the picture of the eye. Okay, so the 2 structures are the sclera and the cornea. Okay, the sclera is the white part of your eye. It's made up of, this should be familiar to you, it's made up of dense, regular, fibrous connective tissue. Tell me something about that tissue. Has a ton of collagen fibers that all run parallel, so what characteristic does it give an organ? Strength. You find a lot of it in ligaments and in tendons, but you also find it in the sclera. And so it makes for a very tough, protective outer part of the eye. So all of this is the sclera. Okay? It's about 5/6, I think your book says, of the outer surface of the eye. It's all but at the very front. And then at the back, it becomes continuous with the fibrous covering of that optic nerve. Okay, so that's the sclera. In lab, you're going to appreciate the sclera when we do this next week, because you're going to have to cut through it. Even though it's thin, it's very tough. It's hard to cut through. Right? At the front of the eye, this part, that's the clear window of the eye called the cornea. You see the cornea? So it's got to be clear because that's what light waves pass through. It doesn't have any blood vessels in it. If it did, it wouldn't be clear, and that would block light waves. Okay, so it's perfectly clear. So this is that area I was referring to a minute ago that gets its oxygen directly from the atmosphere. So that oxygen has to be dissolved in water. So even if you wear contacts, some of your tears still can get on the underside of the contact. So it's still wet there. And if you look at the fine print on your little contact boxes, it probably says somewhere on there, gas permeable. A lot of contacts are gas permeable. So the oxygen can get through. But even if they're not, it still can get through with the liquid, dissolved in the liquid going up underneath. So, okay, so that's the outer layer of the eye. Okay, if you go back a couple of slides, you'll see the middle layer of the eye has several parts. I don't know why my eraser's not working. Here we go. There. All right. If you look at the middle layer, the first thing listed is the choroid coat, right here, the choroid coat. And this is it. It's on this picture, it's looking kind of red, but in a real eye, it's going to be pitch black because it is loaded with, what does it say in your notes? What is it loaded with? Melanocytes. Where else have you seen melanocytes in A&P? Please know this. Where? In the skin. And it produces that brown pigment called melanin that gives your skin color, that protects it from ultraviolet light. Well, you also have a whole lot of melanin production at the choroid. And so as light waves come into the eye, any excess are absorbed, because that's what these pigments do. They absorb light. So it doesn't just bounce around all the time in the back of the eye. So that's the choroid. It's also very vascular. What does that mean? Loaded with blood vessels. So this is a nourishing layer of the eye. Okay. Towards the front, the choroid gives rise to a smooth muscle. There you see it right there. Now this is just on section. It actually forms a ring all around this structure, which is the lens. Okay, and this muscle around it is called the ciliary body. And if you look on this picture, you can see these tiny little suspensory ligaments that hang on to the lens. And so this ciliary body is continually pulling on those little suspensory ligaments and letting go of them a little bit so that they get taut or not. And so that, do you see how that's pulling on the lens? So sometimes the lens gets thin, sometimes it gets thick. And as it changes in its shape, that bends a light wave a little bit differently. Remember I said light waves bend as they go through these different areas of optical density, different media. All right, and so when you're looking at something close up, the lens has to get real thick in order for you to see it clearly. But when you look at distant objects, the lens thins. And all that thickening and thinning is the result of the action of the muscles in the ciliary body. Okay? Because the lens is very elastic, has elastic fibers in it, you tend to lose that ability of elasticity as you get older. Just like you wrinkle, because elastic fibers lose elasticity in your skin. That's one reason you wrinkle. Your vocal cords lose some of their elasticity, so your voice changes. An elderly person's voice is very different, isn't it? You have more cardiovascular issues as you get older, and one reason is because your blood vessels are losing elasticity. Well, you also have trouble accommodating. That's the ability of the lens to change shape. It's called accommodation. That's in your notes down the road. But you lose that ability as you get older, and kind of the magic age is 40. And at that point, people can't see things up close as well. So what do they start wearing? Reading glasses. And it just kind of gets worse and worse and worse and worse and worse. But it's because the lens loses its ability to accommodate. Okay, and so we've done 3 of the 4 structures of the middle layer. We've done the choroid. That's the really dark layer. We've done the ciliary body, and that is muscle that surrounds the lens. We've done the lens. And then in front of the lens is another muscular structure that extends from the ciliary body. You can see it right here. And it is another ring-like muscular structure, but it's in front of the lens, not around the lens. And it's called the iris. And it has various pigments in it, and that's what gives your eyes the color that they are, whether they're brown or blue or green or somewhere in between all of that. And that's just a genetically controlled thing. But the purpose of the iris is to control how much light is able to get through. You know, there's a hole in the middle of it. See this hole right there? That's the pupil. And sometimes the pupil is really tiny. That means it's constricted. Sometimes it's really big. That means it's dilated. And so it gets constricted when you're experiencing areas of bright light, doesn't it? And it gets very dilated when you're in dim light to let light come on through. The iris controls that. Okay, so we've done the outer layer made up of the choroid and the sclera. The middle layer made up of, I said that wrong. The outer layer made up of the cornea and the sclera. The middle layer made up of the choroid, ciliary body, iris, and lens. And then the inner layer of the eye is where the photoreceptors are. It's completely nervous tissue, and it's called the retina. And the retina lines the area where the choroid is. So that's what you see, this yellowish structure right here. That's the retina. And so do you see you have it everywhere at the back of the eye? And all of this is all this sharp part of the vision is part of the retina. Okay, and you have it everywhere except that little part right there. And that part that I have circled is the area that's penetrated by the optic nerve. It's called the optic disc. So in that one area at the back of the eye, you don't have any photoreceptors. So sometimes we call that your eye's blind spot. So if a light wave hits it, you're not going to see it. Okay. But because it's not directly at the back, it's angled medially a little bit. So here would be the blind spot on one eye. The blind spot on the other eye is a little bit, it's the opposite direction. So light waves that are hitting this area on this eye would actually be hitting here on the other eye. So you're still going to be able to see it. Your brain interprets it fine. So as long as you have vision with both eyes. Okay. So here's just some other views of some of these structures. Like at the front of the eye, you can see the lens a little better. The ciliary body's suspensory ligaments that hold it in place. You can see the iris. That's a good view of the iris and the pupil. Pupil's the opening in the middle of the iris. But what I've not talked about are the fluids. Okay, the fluids fill the cavities of the eye. The big cavity at the back is called the posterior cavity. So that would be this area is the posterior cavity. And it's filled with a fluid that's jelly-like, which is called the vitreous humor. Okay. The vitreous humor. Okay. And then the anterior cavity is everything in front of that. So let's see if I can erase some of this stuff. Well, every time I do that, it just circles it. There. All right, so the anterior cavity is going to be all of this area is anterior cavity. So it's around the lens and in front of the iris, you know, so it's all of that. Your book talks about the anterior cavity being in two compartments. You can see that, the two chambers. You don't have to worry about the chambers for me. Just know anterior cavity, okay? Technically, the anterior chamber is in front of the iris, and the posterior chamber is behind the iris, okay? But the whole thing is filled with a different fluid called the aqueous humor. So that's more watery. Okay. If you hear that someone struggles with glaucoma, you heard about that? That's where they're producing a whole lot of the aqueous humor, which puts a lot of pressure in turn on the vitreous humor, which puts pressure on the retina, which can destroy some photoreceptor cells. If somebody has cataracts, that's when their lens is not perfectly clear. It gets pretty cloudy. So do you see how that would interfere with light waves going through? Okay. All right. I love this picture. Look at the section. Look at the top up there. Do you see this section is just behind where the lens is? And so you're looking out towards the front of the eye. And the reason I like that is because you get the idea of how big the ciliary body actually is. That whole orange ring around it is a ciliary body. Okay? So it's not just one little structure like on those other sections. It looks like one little snip. But it's a whole ring around the lens. Okay. All right. Now, I've talked about the lens changing shape as you look at objects of varying distances. The ability for the lens to do that is referred to as accommodation. Okay? And I need you to know for the test that when you look at something close up, the lens has to thicken. Okay? All right. So the lens thickens when you look at close objects, and the lens thins when you look at more distant objects. Okay? You're not aware of that at all when you're not really aware of it thickening or thinning anytime. But when you're young, you know, it's not a big deal to look at something far away and then look at something close up. And it's just, it all is in focus. But just trust me, as you hit about age 40, you're going to be like in the car, and you're going to be reading a book. You're not driving. This is what happened to me. You're reading a book, so you're looking at something close up, and then when you look up to try to read a road sign, it's going to take a moment for it to get in focus. And it just, that was my first indication that, oh, my accommodation's not working so well. And of course I thought that because I teach this stuff, so I knew what was going on, but I really could experience that. And then later, when I looked at something far away and then tried to read that thing, I couldn't read it at all, and it never would get in focus. So it just tends to lose its ability for that. So, all right, now this view is showing you, if you look at the arrows, that's referring to the aqueous humor that's in the anterior cavity. And so do you see how it's actually coming from the ciliary body? Ciliary body has blood vessels in it, and this all comes from the blood. Body fluids are constantly moving from one compartment to another, to another, to another. So it's in the blood, then the watery compartment is in the anterior cavity. Now it's not called blood plasma, it's called aqueous humor. But then it drains. Do you see this right there? It drains into another blood vessel that encircles the edge of the cornea, and it's called the canal of Schlemm. That's a vein, and so it gets back into the bloodstream. So it's constantly moving different places. If it's not draining properly, then that's going to set up some glaucoma. So when you go to the eye doctor and they do that little spurt of air that's bothersome, how they're checking for the pressure at the front of your eye to see if that pressure is elevating. And if it is, that's an indication you may be getting a little glaucoma. Okay, now I also have talked about the iris action. The iris acts like a shutter on a camera. Just like on your microscopes that we use in lab, you have that iris diaphragm built into the condenser. Okay, the iris of your eye works just like that. So it's constantly contracting, muscles in it are contracting or relaxing to control the size of the pupil. You actually have two groups of muscles in the iris, and you can see those on these pictures. One group is circular, so you see the circular fibers here, and then another group is radial muscle fibers like this. Okay, so I'm going to draw some pictures where these are separated. So I'm just going to put the radial in one picture and the circular in another picture. Okay, do you remember, let's say if here's radial muscles. Okay, so you can see the iris coming into view, right? There's your iris right in the middle. Right there. Okay. When muscles contract, when lengthwise muscles contract, remember how there's an origin end that stays put and an insertion end that moves? Okay, the insertion end is the medial end. And so when this contracts, I'm not sure I can draw it right. Well, my eraser's not working, my online folks. Sorry, I'm messing with this smartboard. It's messing up. All right, look. See how I just contracted those muscles? And so do you see how that widened the opening? Okay. All right, and so that contraction is the result of, what do you think, sympathetic or parasympathetic? Remember your situations I told y'all earlier? Sympathetic is when your pupils dilate. Remember, you're scared in New York City. Remember those situations? Did I go over that? I know I did it back in A&P 1. In A&P 2, let me give you, I know most of y'all have had this from A&P 1, but we have a few new students, and this is for my online students too. I want to give them the sympathetic situation that helps you remember what sympathetic does all over the body, and the parasympathetic situation that helps you remember what parasympathetic does. Okay, your sympathetic situation is you're in New York City, it's 3 a.m., you're in Central Park, and you're all alone. Okay? It's very dark because somebody shot out the street lights. Okay, and so what do your pupils do in darkness? They dilate. So see, the sympathetic situation is they dilate. But this is also sympathetic because you hear a gunshot in the distance, and then you hear screaming, and you hear footsteps getting closer and closer to you. So what's your heart rate going to do? Go up. What's your breathing rate going to do? Go up. You're not going to stop to use the bathroom. You're going to run, aren't you? Okay, so your muscles get a lot of blood flow to them. All right. And the last thing you're going to think about is stopping at the restroom. All right. Because in sympathetic activity, sympathetic impulses slow down digestive activity and urinary activity. Okay? So that's your sympathetic. Parasympathetic is Thanksgiving Day, and you're the guest, and you don't have to cook it or clean it, or you just go and you eat and relax. Okay, so you've just had this big meal, you've had a lot of tea, and you go to the living room and visit with your family or watch TV. So what does your breathing rate do? Slows down. What does your heart rate do? Slows down. Your sweat gland activity slows down. All right, but pretty soon you're going to have to go to the bathroom because urinary activity and digestive activity speed up in response to parasympathetic impulses. And when you go to the bathroom, you turn on a light, don't you? So what do your pupils do? They constrict in bright light, right? They constrict in bright light. Okay, so parasympathetic impulses activate the circular muscles of the iris. So if here's your circular muscles, when a circular muscle contracts, it gets smaller in diameter. Okay, so it goes from that look to this look. So do you see how that pupil, here's a pupil, see how it now is constricted? Because of a parasympathetic impulse. So for this test, you need to know that sympathetic impulses cause the radial muscles to contract, which causes pupillary what? Dilation or constriction? Dilation. And then you need to know that parasympathetic muscles cause the circular muscles of the iris to contract, which causes pupillary constriction. Okay? And it says all that in your notes here. Okay, but sometimes you need to see that. And it's easier to see it if you separate on pictures, you separate the circular from the radial muscles. Okay, now I have talked a lot about eye function already, but I have not talked about the retina and the photoreceptor cells. For once in A&P, a term is easy. It's not some long, complicated Latin word. The photoreceptor cells in your retina are called rods and cones. Okay? Those are the photoreceptors, rods and cones. I don't know that I actually wrote that down. Those are the photoreceptors. Okay, rods and cones. Okay? All right, so if you'll stay on this page, let's look at a picture of this. Okay, what you're looking at are the three layers at the back of the eye. So here's the thick layer. That's the retina. See how it's several layers, but by far mostly neurons, and mostly bipolar neurons. And then there's the choroid, so you'd have all these melanocytes in there. That's the real dark layer. And then here's the sclera, okay, dense regular fibrous tissue. And then here is the that's the posterior cavity. So let's say light waves have come through there. Okay, what the light waves are going to do is going to go in between all of these neurons until it gets to this last layer of neurons right here. There's your layer of rods and cones. So they're at the back of the retina. And you can obviously see a physical difference in a rod and a cone. Okay? And there are pigments in this part that gives it the name rods and cones. This is a cone. Here's pigment molecules. And pigments always absorb light, like chlorophyll in a plant absorbs light, doesn't it? Okay, melanin absorbs light, okay, and reflects that it does not absorb, right? All right, and so you got pigments in the long, skinny rods, and you got pigments in the short, blunt cones. And the difference in the two, there's two or three differences, but one difference is what kind of light wave is absorbed by these different pigments, okay? And if we take rods first, okay, Rods, pretty much any wavelength of light can activate a rod. Okay, whether it's a blue wavelength or a yellow wavelength or a red wavelength. Anyone can activate a rod. But the downside of that is it takes real bright, intense light for a rod to get activated. Okay, so when rods are activated, um, they help you in seeing in dim light, but you can't really specify what color it is. Okay, it's more of your grayscale vision. All right, but the cones, on the other hand, have different kinds of pigment molecules, and they are activated by different wavelengths of light. Okay, so in other words, they allow you to see in color, cones in color. Okay, so that sounds like, oh, well, that's better than a rod, but it takes a lot of light to activate a cone. So they don't function well when you're in dim light. You're mainly using your rods in dim light. Cones function a lot better in bright light. Okay. All right. Then another difference in the two. I want you, if you'll look up here, this is pretty obvious. Let's say this rod is activated by a wavelength of light. When it's activated, a nerve impulse is then transmitted. Look, along this little pathway from one neuron to the next to whatever vision area point it is in the brain. All right, but what if when it gets to the brain, because this pathway would be the same as if this one was activated or this one, your brain doesn't know which of these three it came from. So in other words, it's going to be kind of fuzzy. It's not going to be sharply focused because all three of these share a common pathway to the brain. Whereas if you look at a cone, do you see how each of these cones has its own unique all by itself pathway to the brain? So when it gets to the brain interprets it as coming from exactly that spot and not right next to it. So that means it gives you sharp focus. So the good thing about the cones, there's two good things. Color and visual acuity, which means sharply focused. Okay, and it says that in your notes on the page before this. But the downside, what's the downside of cones? Of cones, it takes bright light. You have to be in bright light for them to be useful. Okay? The good thing about the rods is that it helps you see at night or in dim light. They help with your night vision. The downside of the rods is that it's not in color and not sharply focused. Okay? Okay, now what I want to do, I want to turn off some lights in this room. So if you're at home viewing this recording, you can just do what I'm say that I'm doing in this room. All right, right now it's very brightly lit in the room. So I want everybody to look around and see all the nice little cute faces, sharply in focus, and you can see all the bright colors, can't you? All right, what's being activated? Cones or rods? Cones, because it's bright light. Now, I'm going to turn off the lights. Now, we still have the projector light on, but look how people's faces are not as clearly in focus, especially if they're far away from the projector bulb. And the colors aren't as bright, are they? So this is more your grayscale vision kicking in. So that would be your rods. Right, your rods. That's pretty neat, isn't it? Yep. Have you ever heard that dogs can't see in color? How do they know that? How in the world do they know how a dog sees? Because they have gone into the retina of a dog's eye, looked at it under the microscope, which you're going to do. We have prepared slides of the eye and in lab, and you're going to see the retina, and you can actually pick out a rod and a cone. They look different. And I'm not going to say that dogs don't have any cones, but they have a lot fewer than we do. And so they can't see your face in sharp focus. But they do pretty good at night. Okay? So you don't necessarily have to leave light on for them. They see a lot better at night than we do. Good question. Colorblind people are missing one or more of the different kinds of pigments in their cones. So they can see some colors, but not all colors. Some animals have a lot higher concentration of cones than we do. Have you ever heard the phrase, You got eagle eyes, or vision as sharp as an eagle. All right. Eagles and hawks and birds of prey, they're flying up real high, but they can see that little mouse or whatever they're trying to catch, little snake, hundreds of feet away that we may not be able to see because they've got much better sharply focused vision than we do. A chicken has a lot more cones than we do, believe it or not. A chicken can see that little worm in the grass long way away that we would never be able to see. But chickens are kind of blind at night because they have so many cones, they have very few rods. So that's why you don't see them out at night. But here's the real thing. Through the microscope, what you're looking at is the back of an eye. And so you see the sclera. And look at the choroid. See how dark it is? That's all those melanocytes. And then the rest of this is retina. So the retina is actually thicker than you realize. The layer of rods and cones is right there. And this is not quite as magnified enough to really distinguish between rods and cones. But if you stand back away from this, you'll notice that some of these are just a little bolder than others. Do you see that? Like there and there. Those are the cones. And the rods are real skinny. Okay. On a higher view, you can see it a little bit better. Okay. I've talked about the bending of the light, and I wish I had I need to go. Let me go back to a different picture before we get to that bending. I want to show you something. This picture. Oh, that's not going to do. Now. Don't know why this sensor is messing up. Okay. Still doing it? Oh, it is. I'm picking up this eraser, online students, and if I circle it, it should erase everything. And it did not. Let's just have to do it this way. All right, I won't erase all of this, but catch some of it here. Okay, let's get back to the very beginning of this lecture on the eye. And remember I said a whole point when you're looking at something straight ahead, looking at it, focusing directly on something, you want it to hit a particular spot in the back of the eye. Okay, that spot you want it to hit is called the fovea centralis, which is right in the center of this entire depressed area called the macula lutea. For your test, I will not use the term macula lutea because I don't want you to confuse it with the macula of the inner ear. That's for static equilibrium. Okay? But in the dead center of that area is your area of sharpest vision, and it's called the fovea centralis. And the reason it's the area of sharpest vision is because it's all cones. And the farther away you get from it in each direction, the fewer cones and the more rods. So that's why when light waves are hitting here or here, that would be from your peripheral vision, not what you're looking dead at. You don't see that as in sharper focus, okay? And you don't see the colors as well. It's a little bit blurry. Okay? Tonight or early in the morning when it's dark outside, I would love it if you would go out and look up at the stars and just start enjoying the stars, and you'll notice in your peripheral vision there might be a super dim star. But as soon as you turn your head and look straight on at that, it disappears. Tell me what just happened. Think. When it's in your peripheral vision, it's hitting over here, right? Remember I said it was a dim star? So it's activating the rods that are over there. But as soon as you look at it, it's so dim, it's not enough intensity to activate that cone right there. And it really will disappear. It's like magic. So you ought to try to do that next time you look at the stars, when you have nothing else to do. Okay. All right. Okay, so now we're going to look at this light refraction. Refraction is the physics name of the bending of light as it passes through areas of different optical density. Air has one optical density. The cornea has a different optical density. The next structure that the light encounters then as it goes through the cornea is the aqueous humor, which has a different optical density. Then it's going through that hole, the pupil, to hit the what structure? The lens. The lens, which has another optical density. And then after the lens, it gets into the vitreous humor, which has another optical density. So it's bending, 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 bending as it makes its way back to the retina. Okay, and that bending is called refraction. Okay, um, and and if we look, we'll come back to some of this stuff. Look at this. On the left, you can see that when light hits a convex surface, see how it converges? Those light waves are converging to one little spot. So that gives you the idea why the cornea is rounded like it is. Okay, it's convex. If it hits a concave surface, see how it's going to diverge? Typically, when it's hitting a solid surface, the light waves bend so it's perpendicular to the plane of that surface. If you're in a physics class, you'll go over some things like that probably one day. But this is pretty cool. In the middle picture, you can see a normally shaped eye of a person who has the classic normal 20/20 vision. You hear about 20/20 vision. In lab, we'll explain what that means. But do you notice that those light waves that he's looking straight at, they all get angled right at the point of focus, which is the fovea centralis. See that? But look at the eye at the top. Do you see how it's a little bit longer? And so as those light waves bend, they come together before it hits the retina. And so as they come together, they just kind of keep on going, and see how not all of them hit the fovea centralis? All right, when the eye's too long, that's called myopia, and that gives you the vision called nearsightedness. You can see things up close but not far away. Okay? On the other hand, when your eye's too short, by the time the light waves get to the retina, they haven't completely converged yet, have they? So again, it's blurry, and that's called hyperopia when the eye's too short, and that gives you the vision condition called farsightedness. You can see things far away but not close up. You also get that because of lens losing its accommodation, but you can get farsightedness in different ways. One is the shape of the eyeball being too short, and the other has to do with the lens not thickening when you look at something close up. But anyway, corrective lenses like glasses and contacts fix the eyeball being too long or too short. All right, and so if a light wave hit a concave surface, it's going to cause divergence a little bit. See how the light waves have come out? I'm being drastic on this. It's come out a little bit as they hit it. And so that they're farther apart, so that when they come together, it'll be just at the right spot. Okay? All right, and then if the eyeball is too short, the lens's shape is different, so it makes it, instead of coming in like this, it's going to come in a little closer. So it will converge at just the right point. Okay? How many of you wear glasses? And I know contacts are doing the same thing, you just may not realize it. There's a curve on the contacts. But when you were little, how many of you started wearing glasses when you were like in elementary school? Okay. Back then, did your prescription change yearly? But then as you get older, it doesn't seem to change as much, as frequently. And that's because your eye has reached its adult length. See, it's changing shape as you get older. Okay. If somebody has an astigmatism, a lot of people have to wear glasses and contacts because of that, and that's usually has to do with the cornea, or maybe it could be the lens, but it's not perfectly shaped. So it's not the eyeball being too long or too short. It's because the cornea's not perfectly shaped. Let's say here, there's an astigmatism. Let's say this is out a little bit. It may converge at the right length, but it may be up here or there, not right in the dead center of the fovea centralis. Okay, and so again, corrective lenses can fix the curve. Okay, when somebody has LASIK surgery, they're changing the curve of the cornea to fix how the light's going to refract, so it will get to the right spot correctly. So, interesting. Okay. Okay, now this is neat. I want you to just analyze this picture a little bit. Do you see? He's looking at this tree. But by the time the light wave that's coming from the top of the tree gets to the retina, it's at the bottom. And anything on the left is going to be on the right. So see how the image is totally reversed? The reason you don't see it upside down, perceive it as being upside down, is because by the time the impulses get back to the vision center of your brain, it has righted itself. So everything looks upside down there. Okay. Now if we look at vision chemistry with the rods and the cones, this is not heavy chemistry, it's easy chemistry. The pigment that gets activated by light waves in your rods is called rhodopsin. And when a light wave hits it, rhodopsin is going to break down. And when it breaks down, that releases some energy, which is then used to fuel the action potential. But as it breaks down, it breaks down into a protein and then one called retinine. But in order to have adequate retinine, you need vitamin A. Vitamin A is very important it's very abundant in some of the foods that you eat, like carrots. Have you ever heard that you need to eat your carrots? If you don't eat enough carrots, you'll have night blindness. Okay, that's in a way that's true. If you don't get enough vitamin A in your diet, regardless of where it's coming from, if you don't get enough vitamin A, you won't be able to build enough rhodopsin, and it will affect the action of your rods. Okay. Some of the advertisements for vitamins these days, especially for vitamins for mature people, they say they contain vitamins for the eye. They're talking about vitamin A and some others. But as you get older, you have more of a need for those vitamins. Have you ever heard of a condition, an eye condition that some people struggle with, and it can get real serious? It's called macular degeneration. The macula is that area around the fovea centralis, and sometimes the cells there can start breaking down, and it can cause permanent blindness. Sometimes people just have little mild versions of it, but they need to take these vitamins. They need to get enough of the vision vitamins, and vitamin A is the big one. Okay, because rods get activated by even tiny intensities of light. During the day, can you see that they're all broken down at one time? They're just pretty much overwhelmed. And so you don't have any left at that moment. So that's why your cones pretty much take over during the day. Right. But you probably noticed if you're in dim light for a long time, and then all of a sudden you get in an area of real bright light, you're temporarily blinded. And that's because all of those all of the rods are breaking down at one time. So it takes a moment for the cones to kick in. Okay. There are 3 different visual pigments in your cones. One is sensitive to the red wavelength. Your book names these. You don't have to name them for me. But one sensitive to the red wavelength, one to the green, and one to the blue. Now you notice those aren't what we think of as our primary colors. I think, I may be wrong, are any of you are y'all really into art? You've had a lot of art classes. All right. Are red, green, and blue primary colors for absorption maybe? And then red, yellow, and blue I think maybe primary reflection colors. I don't know. But anyway, the combination of what gets absorbed. what light waves get absorbed determines the color that your brain actually perceives. Okay. Someone a minute ago mentioned about color blindness. If you are missing this pigment, then the blue wavelength's not going to get activated, or the blue pigment won't get activated. So you'll have issues with some of the colors that you perceive. Okay. Okay. The optic nerve, I know it's in your notes somewhere, but you need to know what an optic disc is. The optic disc is where the optic nerve penetrates the back of the eye. So just right where it's penetrating, just right there, is the optic disc. And because that's where the optic nerve is, and the nerve fibers just extend out onto the retina, you don't have any rods or cones there. And so that's the eye's blind spot, okay, at the optic disc. It's somewhere in your notes, okay. All right, but I want you to notice on this picture, look at the eye on the left. Do you see how it's got the little blue lines coming from it? Those represent some of the nerve fibers in the optic nerve. And do you see how some of them cross over at the optic chiasm and some of them don't? So some of the images from the left, or some of the impulses coming from the left eye, are going to end up on the right visual cortex of the brain, but others end up on the left visual cortex. So both sides of the brain get activated by each eye. There's a crossover area there. And so that's why, if you lose vision in one eye, you still can see pretty well. What you're losing, let's say if you lost vision here, you're just losing some of the peripheral vision from that left eye. But so we have what we call stereoscopic vision, because both sides of our brain are activated by each individual eye. Okay. All right. We're going to take a break here, and when we get back, we will start the next chapter.